this one is also used. So it all starts with this. So starting from the top, again, I'm not recommending this, but this is a mnemonic you could use to help you remember. So it's come, let's get sun good, right? I want to get sun good, it's not pleasant, it's not good, right? Those just what let's help remind ourselves of first letter. It all starts with stratum, which means layer. And this is going from the outermost to the innermost. Right? This is the one that's different. Couldn't find it. So we want to okay, so the top one is called cornea. Stratum cornea. Okay. The next one is stratum lucidum. Next one is granulosum. Next one is spinosum. And the last one, basale. In terms of cells, they are going in this direction. They start here and migrate, 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 and what's sloughing off is from stratum. Okay, just realize that. We start with the cells, like we start with basale and then work the way up. Okay. Having said that, so stratum basale is where we have mitotically active cells. Cells are divided and pushes upwards. Spinosum, here, these keratinocytes, that's because of its name given to the cells. Those are the most prominent cells in our skin. Yes, you may have heard of other ones which produce melanin, melanocytes, but that's not the most prominent. We have others too. The most prominent is the keratinocytes, right? So here, it's spiny keratinocytes. So they got this porcupine, almost looks like a pufferfish or a porcupine, where they have these spiny appearance. So that's the functional significance or the historical significance of granulosum. They have a lot of granules. So keratin granules are the highest here, or they're producing a lot of keratin granules. Okay, so spinosum, spiny keratinocytes. Granulosum, because there are a lot of these keratin granules that are it's the darkest in terms of layer. Lucidum, not always found. I always suggest remembering Lucifer, okay, to help us remember what it does or kind of relative to it. This is where cells die, start to die, okay? And then corneum is all dead cells. That's what we're shedding, okay? Are we shedding a lot of skin cells?
Yeah.
Yeah, I'll tell you how much in a second. Okay?
You did already.
I did. Yeah.
No, I don't see my list.
Tens of thousands. It's 50,000 a minute. Okay, that's a lot of cells. Okay, that's coming from here. That's the dander in our house from all the dust mostly is from those dead cells. So cells are migrating from the bottom up, right? What needs to be close by here? These are mitotically active cells. Blood vessels, right? So these will be where the connective tissue is. What should we label here then? Dermis, right? So it's not papillary layer of dermis. Top part dermis. This is epidermis, the epidermal cells, because the mitotic active has to be closest to that. these are dividing from here, they're pushing up, cells are migrating outwards, right, more superficially. So it's stratum basale or germina vitum, that's one of the book talks about it, whether it's germinating, they're multiplying. But in terms of mnemonic, I couldn't find one. I prefer this. As far as stem cells, they're dividing. So here is to show the dermis, the pinkish one, blood vessels, all of this. How we try to identify them, just know which order they are and what's the function. That's all structures. Here, most mitotic cells. Then the spinosum, spiny granulocytes, so that we have little spines-like shape on their cell membrane. Stratum granulosum, lots and lots of keratin granules. And then lucidum, this is where cells are dying, and beyond that, you just notice that is quite a thick layer of this. Very realistic in terms of how thick it is, up to about 40 layers. These flattened dead cells, so we're losing them, but we're protecting. What's the functional significance of stratified squamous epithelium? Yeah, so we have all these cells which can be sloughed up, we don't care about them because they're dead cells. They actually have lost everything, we've claimed it back. There's no nucleus, there are no organelles virtually, just thickened old keratinocytes. So cell differentiation, it takes roughly about 2 to 4 weeks to go from that basal layer all the way to the top of cornea. Okay? Unless we suffer with any other disorders such as psoriasis, this process is much quicker. Okay, but generally it takes about 4 to 6 weeks. At the top, we talk about lucidum being where there's cell death. Apoptosis is 2 ways to think of it. It's like programmed cell death, or these cells are dying suicide, cellular suicide. But it's controlled, right? We want these to be dead cells, but we want to not have to lose the other cells. So what happens to them? Pretty much all the organelles are broken down, the membrane thickens again, trying to protect the underlying layers, right? And these break off either as dandruff, dander, and so on, right? We lose quite a lot every minute. About 50,000 cells are lost, shed in. Any questions? Going from the bottom up is how the cells work. Okay. But the acronym kind of helps us. Sun is outside, helps at least get the first letter. So the reason for difference in our skin color is 3 main types of pigments. Okay, one is melanin, of course. And we also have 2 forms of melanin, right? We have the brownish black melanin and we have the reddish orange melanin. Okay? So the reason again difference can be due to the amount and which form of melanin we have. The form is generally determined, but how much we have can vary, right? You might have noticed amongst yourselves, summertime versus wintertime. Does your skin color change? Yeah, right. It does, so that the amount does change, but what we have is generally determined, right? What kind of keratin, more seen in the creases of our palms and feet, right? What does it sound like? A vegetable? Carrots. What color do you think it might be? Orange. It actually gives us an orange tinge. You don't see it very often except for in certain places or dimensions. The other one is our hemoglobin. Where does normally hemoglobin reside?
Red blood cells.
Red blood cells. What's the function of hemoglobin?
Carry oxygen.
Talk about that's where 98% of our oxygen is carried bound to CO2. It can carry CO2, but not as much, right? Keratin, what's the reason or what's the function is that it will convert, it is actually converted to vitamin A, which is required for good visual health. Okay. They also added into creams to what's good for skin health as well. Thankfully, it's not very common, but vitamin A deficiency can lead to blind consumption. In the US, at least, it's not common, but still in some countries it is still common. Vitamin consumed in the diet. Question? Okay. Moving on, let's talk about skin and UV radiation. So, what are some things that UV radiation does? First of all, we talked about vitamin D. Okay, skins produce it with exposure to sun, right? But we don't want the UV part of it, okay? Because that can cause skin cancers. That's the number one cause of skin cancers, right? Nothing else comes even close to it. Okay? Others do trigger, but skin cancer is the number one cause. Is the UV radiation. What they do is they change either the bonds between nucleotides or during cell division, they will change the nucleotides in our DNA, hence that can cause cancers, right? Not necessarily, but can cause cancers. Can also cause premature wrinkling. If you see someone who has done a lot of sunbathing in their earlier life, roughly in the 50s, 60s, you might see wrinkling. Somebody who works outdoors, you'll see more wrinkling again. It's in more sun-exposed areas because the UV radiation also does increase wrinkle formation. So let's talk about some skin cancers then. So 3 main types. Okay, so we have the squamous cell carcinoma, basal cell carcinoma, and then the other one? Melanoma, right? Which is the most dangerous? Why is it the most dangerous? 2 big reasons. Why is it dangerous? One, what is the... So is it problem here? The melanoma's right here? No, when does it become a problem? Or when it becomes dangerous? Yeah, it can spread anywhere. It can metastasize, especially to the lungs, the brain. You can feel that too. That's one thing it's dangerous, right? Why is it another? Well, the other 2, they can. Well, this one can metastasize, not so much. The basal cell does not, but. Oh, actually, I forget that. Yeah. Why is another reason why melanoma can be dangerous? It moves, it grows, it does grow quickly. It also how many people have moles? Yeah, pretty much all of us do, right? It can resemble very similar to a mole when it starts off, right? And then we don't realize it could be a melanoma. Now, talk about some things that we can look for. You've probably heard of it too, but there are things that we can try to look for because it looks like a normal. All the other 2 does not look like normal. You will see it. If you've seen, I'll show you pictures. It looks normal. Whereas moles, as we saw, pretty much all of us have it. Melanomas can look like moles. Right? Which is why if you stay there and you don't realize. Okay. So, basal cell carcinoma basically comes from where the stem cells are. That's why it's named after basal. It's a basal layer. Okay. Squamous cell. Why is it called squamous cell? What's our guess? Yeah, it is. What's our epidermis made up of? Stratified squamous epidermis, basically named after it, right? So let's look at these. The most common actually is basal cell carcinoma. We get more of it, but it's less dangerous. It doesn't metastasize and it's very rarely. It's not. It is malignant, does spread, but doesn't metastasize. Doesn't go to other organs. go beyond. What's the difference between benign and malignant? Yeah, the board. Spread to different organ, is that malignant and not benign? Spread where? Tissue, yeah, if it spreads to a different tissue, it's still malignant. If it spreads to a different organ, it is metastatic. That's metastasis. So, yeah, benign stays in that same tissue, malignant goes beyond the tissue, metastasis spreads to others. So even though basal cell carcinoma is still malignant, it just does not spread. It's not metastatic, that's why it's not dangerous. While a squamous cell can metastatize, but again, you see one, you will see why it's very easy to catch them. Okay? While melanoma will start like a mole, that we all have, we not recognize them. Here is the melanocytes that are dividing uncontrollably, but melanoma, it does metastatize. Okay? But if it caught early, is it curable? Yes. Yeah, it is. Okay? But these two spread through I think I might have it later on. Let's talk about next one is the dermis, right? So it's got connective tissue. What's the top layer of the dermis called? Upper layer, next layer down. Reticular layer of the dermis, right? So if you have any leather shoes, you wear leather jackets, your wallets, right? What we get is basically we take off all of this. It's the fibers that are left basically within our dermis that becomes the hide part of it, leather part. There's a lot of process treating them, but that's because of the high concentration of fibers in our dermis, right? Again, it has a lot of blood vessels, right? So will I bleed if I only take off the epidermis? There may be some times you might notice you scraped it, but if you not that time you will need to off the epidermis, not go into the epidermis, right? So if we go beyond this, we'll start bleeding if we stay with this. Right? Dermis has a lot of good blood supply. We'll talk about this. Our hair follicles, our nails, they're really epidermal cells, but they're growing from, say, migrated there, and then they're dividing. I know we say that nails are growing from here, but where are they really growing from? Same thing from the hair, we say it's growing. It's really growing from the base, it's just pushing upwards, right? It's the same as this, dividing from bottom up and then pushing upwards. So they contain, these are epidermal structures, epidermal cells that have migrated into the dermis. Two layers, papillary layer and reticular layer. Papillary layer is on top, which is composed of areolar connective tissue, roughly about 20% in terms of thickness, 80% is right. And then what would be below, where these adipocytes are? That'll be a part of our adipocytes. The dermal papillae, we didn't talk about it in lab, but in the model on one side, I think it was, we're looking at it, it's on the right hand side, the whole epidermis is taken up, and then you may have noticed there were these finger-like projections, right? Those are the dermal papillae. Why do we have those instead? Why not have it flat? We think about that. Why do we have these bumps in the dermis rather than being flat in the epidermis?
Increasing the surface area.
Increase the surface area for multiple reasons. You can have more structures within it, more blood vessels, more receptors. It also helps to attach the epidermis onto the dermis. Okay. The dermal papillae are found within the papillae. Those are the little finger-like projections of the dermis. In certain areas, these dermal papillae will fuse, okay, together with the epidermis, and we form these ridges, which are what makes up our fingerprints. They're found in our feet too. Okay, that's fused dermal papillae, and on top, also the epidermis forms a nice ridge on top. Any two fingerprints alike?
Yeah.
No, even light twins, their fingerprints are different. So it's genetic and it's how, in terms of there's some environmental conditions that led to our difference. Lower down is the reticular layer of dermis. Reticular layer mostly made up of dense, irregular connective tissue, but we also have a little bit of adipose connective tissue, and there are larger blood vessels there. So, if you are getting a tattoo or have gotten it, where was the ink placed? Should we place it in here? Why not? It'll be easy. Just put it.
It'll fall out.
Yeah, it's gonna disappear in about 2 weeks, maybe 4. Right? Why should we not put it in the hypodermis?
You'll never see it.
Yeah, it's gonna be very blurry, right? Fat does not allow those nice colors to show up, right? Generally, we put it just a little bit below the papillary. It's the reticular layer that those things in, right? So they're there for longer. Yes, and we put in the papillary layer. It's easier to get to, but the color will dissipate much more faster compared to the reticular layer. But we definitely want to put it in the hypodermis or the epidermis. Epidermis will disappear within a couple of weeks. The other one is coloration. Okay, so next we're going to talk a little bit about the different extra structures of the So, hair follicles, nails, sweat glands, and sebaceous glands. Right? So let's look at the hair follicles. So they're also pretty much dead cells. And dead keratinized cells. Here we have more keratin than we had. What do you think about nails? Even hardened keratin, right? So that's also keratinized as well. Okay. Not found in certain places, you probably can guess. You've seen it in yourself, like palms, soles, on our lips, on the nipples themselves, around the spine. Please note they are epidermal cells that have gone to the dermis and then going out of the dermis. So when we go into, so we have the hair shaft, which is outside. So hair follicles, the basic, all of it. Hair shaft outside, and then we have hair root. At the bottom of the hair root, it widens out to form a hair bulb, right? A lot of the structures, a lot of the activity is within that, okay? So root hair plexus, it covers this hair bulb, the touch receptors. How are they sensitized? When, yeah, the movement of the hair follicles themselves, the hair shaft, right? Right inside this bulb, we have what's called the hair matrix. That's the equivalent of the basal cells. That's where the mitotically active cells are found. Okay? Everywhere else besides that's basically dead keratinocytes that's coming out. We do not have these layers. Middle layer where they're dying, but hair matrix is the one that's the actively mitotic cells. The rectopili muscle we talked about is smooth muscle, right? Just cause goosebumps, it traps air, so we have this layer of air which acts like another layer of clothing, if you will. It protects against the outside cold. Hair papillae, this is within the hair bulb. So if it's epidermal tissue, does it have its own blood supply? No, so it depends on dermis for its blood supply. So dermal papillae, hair papillae is basically dermal tissue that's in the hair. I have a picture, I'll show you in a second. It's within that hair bulb that helps and touches basically or comes very close contact hair matrix. What was the hair matrix? Mitotically active cells, right? That's where division takes place. That's what needs the most blood supply. Okay. So here's the image. So hair shaft, what's outside, hair root underneath or within the skin, hair bulb or root bulb is the widened up base of it. Here is an enlargement of that. We'll talk about that in 1 second. So erector pili muscle is attached to the root. In contrast, you pull the hair shaft straight up. So here, the red cells are basically epidermal cells. The blue is the dermal cells. Okay. So here would be the hair matrix. That makes this the hair pathway, right where it's dermal tissue that's supplying nutrients to the hair matrix. Hair matrix is going to keep dividing. It's going to push the cells out. So, if we want permanent hair removal, what do we need to remove?
Laser.
Do we just remove the hair follicle?
No.
No. What do we want? What part? And what am I looking for? You have to remove this, right? So if we shave, what part of this hair follicle are we removing? Just the shaft, right? If we did some waxing, what are we removing now? A lot of times we do remove some root. We do remove root. A lot of times we remove the bulb intact, but sometimes we leave it behind. Okay. If somebody who keeps waxing might have noticed after a few years, sometimes hair does not grow in that area. Okay. But sometimes it still continues to grow. If it continues to grow, there's 2 reasons. One, we didn't get the hair matrix, or we left this tract intact where some of these cells migrated down. Okay. If we want more permanent, the newer ones is where we do laser treatment. We actually will burn, cauterize this area, thereby causing scar tissue, thereby making sure this is dead, and also no migration of the epidermal tissue down that tract. Right? So, scar. Question? So, yes, I know we're going to push through these things. We'll finish this step and feel a little bit of confusion. So, glands, we'll talk about 3 glands in dermis, but please remember these are epidermal cells, right? I forget which order, but so sebaceous glands. What do the sebaceous glands do? Produce the oil, or it's called sebum, which is our natural moisturizer. If we saw with acne, it's because these glands are blocked and then they get inflamed. That's why you can also see pus coming out because of the inflammation of these glands, right? These glands are attached to the root hair root, right? Root of the hair, so these secretions actually will come into that tract and then the sudoriferous is another name given to the sweat glands. Okay? What was the sweat glands that helps with to cool down our body? Eccrine sweat glands. And I mentioned the other one in the lab, but I said we'll talk about it in lectures. So eccrine sweat glands, eccrine sudoriferous glands, those are the ones that are used for thermoregulation. We do have another type of sweat gland called apocrine sweat glands or apocrine. They are not found all over our body. The most concentrated, the armpits and groin region. Okay? That's where we have apocrine, whereas eccrine spread throughout our body. That's for cooling. Here, it's the pheromones which become body odor and we do not have too much of it or have left it at all. The last one is ceruminous glands. These are only found in certain areas, which is our ear canal. We didn't realize, yes, inside our ear canal too, up to the eardrum, is lined with skin. Okay, so these ceruminous glands produce the earwax that you and I may have known about, right? Is it producing all the time? Yeah, our earwax is being produced all the time. The idea is to waterproof our our ear canal. So there's no water because we want what other thing to travel through? Sound, right? So if there's water in there, that's not going to help sound travel as well, right? So these are present, but generally it comes out little by little. You may not notice it unless you, when you're after a shower, use a earbud, or you wipe it down, you may notice wax. Otherwise, it just gently comes out. Because we're not producing a lot. The reason we may notice collection of earwax is that gentle the process of coming out is not happening, right? Usually common in the young, because the shape of their ear canal, or as we age, because it gets drier more, it gets stuck more. Anybody who suffers ear infection probably has now high chance of earwax that collects. So the idea is to waterproof the ear canal, take anything out of the cerumen infection. Okay, so nails. So again, it is epidermal cells that have gone in. It's right at the base nail matrix. That's where it's growing. It's not growing from here. It's really growing from there, pushing the cells